And thank you for the opportunity. I'm really excited to be here. Thank you everyone for making it today. I'm Priya Devarajan. I'm currently an instructor working with Dr. Susan Swain at the University of Massachusetts Chan Medical School. And I'm looking forward to beginning my own respiratory immunology research group at Stony Brook Medicine soon. So today I'm going to be talking to you about a story that we published recently about how lung cytotoxic CD4 T cells are generated during infection. So we all know when we say cytotoxic T cells, we think about CD8 T cells. But cytotoxic CD4 T cells are a class 2 restricted, they can mediate class 2 restricted cytotoxicity through perforin and granzyme-mediated lysis of infected cells. And you would ask me, why have class 2 cytotoxicity? We already have CD8 T cells that kill. Well, that's because a lot of viruses, including influenza and SARS-CoV-2, can downregulate class 1 as an immune evasion mechanism. And the more mechanisms the immune system has to clear the virus, the more chances you're going to have at clearing it sooner. Also, additional epitopes will be presented on class 2 in addition to class 1. One thing to keep in mind as we go through the talk is that these cytotoxic CD4 T cells are a late CD4 effector subset. So, for example, in a classical Th1 response during influenza infection, you have these first three of CD4 effectors that are generated, there are Th1s, and then you have the more specialized subsets of CD4s, like cytotoxic CD4 T cells that come from these CD4 effectors. Also, the cytotoxic CD4 T cells generated during infections are tissue resident, in that they're only found in the infected tissue, which during influenza infection is the lungs and the noses. And these cytotoxic CD4 T cells have been implicated recently in protection from various diseases. And we have more and more studies coming out showing the importance of these subsets that have previously been understudied. So we have studies talking about how they protect during HIV and TB, and we and others have shown how they protect during influenza infection. Recently, there's also a spotlight on them because there's this really cool study looking at patients who had long-term tumor remission who had gotten CAR T therapy, and what they found was that these T cells had basically converted to cytotoxic CD4 T cells. However, these cytotoxic CD4 T cells are also pretty potent. They're good at what they do. They're good at directly killing their target cells. So you don't want to have too many of them, which as we go through the talk, you'll see there are so many steps regulating their differentiation development. And we think that's why, is because you don't want them unless you need them. You don't want to have too many of them. Recently, we've seen how they've been correlated with severe disease and lung pathology in human patients. And these are also cells that progress with aging. That is, you see more of these cells coming up in humans as they get older. So, if we are looking at infections and we're thinking about vaccines and how to make vaccines, successful vaccines for these infections, traditionally we think about antibodies, right? That's what we think about as antibody correlates of protection. If we take a step further, we think maybe about T cells, usually with CD8 T cells, because we think about T cells in context of these are the cells that can target internal epitopes of the virus that don't mutate often, like with different strains of influenza virus or with seasonal variation of viruses. But if you were to learn from a successful immune response, like a successful immune response against influenza infection, we start seeing multiple levels of sophistication, where it isn't just antibodies, but it is broader antibodies with broader specificities, neutralizing antibodies that are longer lived. And when you see what is required for this higher level of sophistication, that's when we start seeing the importance of CD4 T cells. You see that CD4 T cell help to CD8 is important for generation of longer-lived stable CD8 memory. You see that CD4 T cells, a specialized subset of CD4 T cells known as T follicular helper cells, help B cells to make these more broader, neutralizing, longer-lived antibodies. And something we're going to be talking about today is about how these cytotoxic CD4 T cells, being a part of the immune response, helps prevent virus evasion which usually are able to evade a lot of these more classic T cell responses and B cell responses. And if you think about immune memory generated, it's not just about having antibody memory in the serum and a step beyond, not just having T cell memory, but having T cell memory in these frontline tissues, in the lungs and the noses, which are going to get first infected with respiratory virus. So if we look at infection, we say influenza infection generates stronger immune memory than influenza vaccines, of course, at the cost of pathology, but it does generate stronger immune memory. And you say, why is this? What can we learn from infection that we can maybe apply to vaccine design? We see broader antibody responses that are longer lived. You see quantitatively higher as well as longer lived T cell responses, both CD4 and CD8. And influenza infections generate memory that is conserved against seasonal variants of the flu, that even if the flu virus changes its HA a little bit, T cells are able to target internal epitopes and preserve immunity, which is what we call heteroceptive immunity. And so we say, okay, why is this? What can we learn from infection? And so what we did in lab was we did a very basic experiment. We gave mouse influenza infection, and we transferred T cells that recognize a nuclear protein of the mouse called fluin P, TCR transgenic T cells, and these T cells express NR77 GFP. So when they see antigen, they will light up green. And this is expression of GFP with time. And what you see is that virus proliferates rapidly, but it reaches a peak later on compared to vaccines. And this peak of antigen recognition is somewhat maintained for some time before it declines rapidly. Whereas immunization, you see there's a short bout of antigen presentation and then a crash that happens. So we tried to ask, what role does the sustained high levels of antigen presentation during infection play? And because we were interested in CD4 T cell responses that we see is important for more of these sophisticated levels of immune response, we asked, does it promote CD4 T cell responses? And when we look at longer antigen presentation and how that's important, we see that specialized subsets of CD4 T cells, like T follicular helper cells, which are TFH, and cytotoxic CD4 T cells are really only generated after 6 days of infection, where during infection, there's still viral antigen persisting, but during infection, during immunization, antigen is already gone after 6 days. So you see these subsets during infection, but you don't really see these robust TFH or TSCTL responses during immunization. And so we asked, okay, so we see these cytotoxic CD4 T cells coming up during infection, not so much so during immunization. We think that the sustained levels of antigen presentation is important. So really, what drives the development of these effector CD4 T cells that is generated as soon as the naive CD4 T cells see antigen after infection? What requires to further drive these initial wave of effectors into their development of more specialized cytotoxic CD4 T cells? And so to ask this question, we did a sequential transfer model where we tried to isolate that first part of the immune response, the early effector response, from this later phase where these more specialized subsets are generated. So what we do is we first we isolate naive OT2 transgenic cells, and we transfer them into B6 mice. We infect these B6 mice with an influenza A virus strain expressing the antigen for these OT2 cells. 6 days later, these naive OT2 cells have gotten activated, have a full round of antigen presentation, and have become fully developed Th1 effectors. We then isolate these Th1 effectors, and then we transfer them to different second host mice to ask, what is it that's required in this second phase that we think when these THCTLs are formed? What is the factors driving their generation? So we use different second host mice depending on the question we ask. as a positive control, we use PR8 OVA infection-matched mice, which means that these are also 6-day infected mice. So these are essentially our positive controls, where 6-day OT2 effectors are going from 6-day infected mice into 6-day infected mice. Or we infect these mice with PR8. So these mice have the influenza infection, but they do not have the antigen for these OT2 cells after day 6. Or as a negative control, we just transfer them to completely uninfected mice. 2 to 3 days later, we ask if THCTLs are generated from these 6-day effectors. And we identify THCTLs by their expression of NKG2CE. We published this earlier, that this marker really nicely helps us define these cytotoxic CD4 T cells in the lungs of infected mice. And 100% of class 2 restricted cytotoxicity is restricted to the CD4 T cells that express NKG2 at this time of infection. So we can use the surface marker to be able to track the development of these CD4 T cells, in more complex adaptive transfer systems. And it helps us get over the problem, because usually in the field, we've had to do perforin staining to demarcate these cells, which is an intracellular stain and kills these cells, and we don't really have good reporters for that. So having the surface marker allows us to reliably track these cells, and we had published that earlier. And what we saw was that if we transferred CD4 effectors into mice, into positive control mice, we saw a good development of THCTLs. However, in mice that had influenza infection but not have antigen, after 6 days, you did not see the development of cytotoxic CD4 T cells. And this correlated to their cytotoxic function as well. So this told us that CD4 effectors, even if they're fully developed Th1 effectors at 6 days, this is pretty long after initial infection, they still needed to see antigen again to develop into these cytotoxic CD4 T cells in the lung. And then we said, okay, antigen is important, but we also see that these cells develop only in the lung. So perhaps is local antigen presentation in the lung important? So to ask that question, we generated 6-day OT2 effectors, as we had done previously. And we then transfer them into different groups of PR8 infection mice, where we transferred in antigen and oval pulsed APCs intranasally, or we transferred in APCs intrasplenically, or we transfer them intravenously, or as a negative control, we transfer in unpulsed APCs. And so this was our way of saying, if we provide antigen presentation in different compartments, does that drive THCTL formation? 14 to 16 hours, we first said, where do these APCs localize if we transfer them intranasally, intravenously, intrasplenically? 3 to 4 days later, we asked if THCTLs were generated from these 6-day effectors, if they saw antigen in the lung versus in the periphery. So what we see is that when we transfer APCs intravenously, we see APCs in the spleen and not in the lung. When we transfer them intrasplenically, we see them in the spleen and not the lung. But when we transfer them intranasally, we see them in the lung and not the spleen. Then if we look for NER77 expression on these OT2s, which indicates where these OT2s are seeing antigen, we see that if we transfer APCs intranasally, the OT2s in the draining lymph node and the lung are seeing antigen. Whereas if we transfer them intravenously, the OT2s in the spleen, draining lymph node, and lung are seeing antigen. Whereas when you transfer them intrasplenically, it's only OT2s in the spleen that see the antigen. And what we found is that THCTLs developed from CD4 effectors only when they saw antigen in the lungs, not when they saw antigen in the spleens before going to the lungs. And in fact, it was pretty interesting to us that it wasn't sufficient if they got intravenous antigen. And so when we try to put that data together of APC localization, NER77 expression, what we interpreted this as was that CD4 effectors that got intravenous antigen were seeing antigen in the periphery before going to the lungs. So even if they saw antigen in the periphery before going to the lungs, it was not sufficient. And they had to see antigen in the lungs again to become cytotoxic CD4 T cells. So we now see that local antigen presentation is important after 6 days of infection. Next, we asked, is there anything else that infection is providing other than just local antigen presentation? So to ask that question, we did the same experiment. We transferred OT2 effectors along with antigen into either purine infection matched mice or uninfected mice. So the only difference between these two groups is the presence or the absence of infection. And we had controls showing that antigen was presented equivalently in the lungs of both of these groups of mice. And we asked for THCTL generated from the 6-day effectors. What we found was that even if we provided antigen after 6 days, the CD4 effectors still needed something else from infection in addition to antigen to become cytotoxic CD4 effectors. And when we think about viral infection, the first thing that comes to us is type 1 interferon. So we asked, is it type 1 interferons? Is that what's important from viral infection? And so to ask that question, we transferred these 6-day OT2 effectors into either infection-matched wild-type mice or into IFNAR knockout mice, which are mice that do not have the type 1 interferon receptor. And what we found was that the absence of IFNAR in the host mice resulted in decreased generation of cytotoxic CD4 T cells. And so this suggested to us that the IFNAR was important in host mice, even though IFNAR was presented on the transferred cells. So this told us that type 1 interferon is an indirect mediator of this effect. The type 1 interferon was inducing something else during influenza infection, which was what was directly important for cytotoxic CD4 T cell development. And as we tried to narrow down what this factor was, because influenza infection induces a whole range of cytokines and interferons and everything. So we tried, okay, how can we narrow this down? So we tried asking, when is this factor from infection present? And so to ask that question, we did the same transfers where we transferred 6-day effectors with the antigen, but into either uninfected mice as a negative control or infection-matched mice as a positive control, or we transfer into mice that are infected on 0 days, 2 days, or 4 days. So in these hosts, 6-day effectors had influenza infection from 0 to 2 days, in these from 2 to 4, and these from 4 to 6. And then we asked if TSCTL were generated from the 6-day effectors. What we found is that this infection factor, whatever it is really present optimally only after 6 days post-infection. So then we said, okay, then what is this infection factor again? And now that we have narrowed it down to the 6-day time point, we did a single-cell RNA-seq analysis of influenza-infected lungs. And what we found was that if we looked at CD4 T cells that had just reached the lungs at the 6 days when they begin to convert to THCTLs, we saw that there was IL-15 induced pathway that was driving a blimp hobbit and a tissue residency program. And this is interesting to us because in a previous paper by Marshall et al. In Journal of Immunology, we'd shown that BLIMP1 drives THCTL differentiation. And so then we asked, is it IL-15? So we did a very simple experiment where we took IFNAR knockout mice, we infected them with influenza, and saw if these IFNAR knockout mice simply had lower levels of IL-15. And the answer was yes. So to then ask if IL-15 was important, let me just give you a quick introduction to how IL-15 is presented. IL-15 is trans-presented to T cells, in that the T cells only express the beta and the gamma receptors, whereas the cells that are presenting IL-15 need to also express the IL-15 or alpha receptor. So if we wanted to ask the question, if IL-15 was important, we transferred these 6-day effectors into either wild-type mice or IL-15 or alpha knockout mice. And we asked if TSCTL were generated from 6-day effectors. And indeed, we did see a decrease in TSCTL generation in these IL-15 or alpha knockout mice. And so then we said, then if we add back factors that are important, which is local antigen presentation and IL-15, can we generate cytotoxic CD4 T cells from 6-day effectors in just an uninfected mouse? And so we transferred 60 effectors along with antigen-APC into our positive controls, which was our infection-matched mice, into our negative controls, uninfected mice, or we transferred them to mice and treated these mice with IL-15 complexes intranasally. And asked for TSCTL were generated from the 60 effectors. And the answer was yes. As long as there was antigen presentation in the lungs after 6 days, and as long as there was IL-15 being presented to these effectors, we could generate cytotoxic CD4 T cells from 6-day effectors. So to summarize, what we see is that we know that naive CD4 T cells get primed to become effector CD4 T cells during influenza infection. However, these effector CD4 T cells need to go to the lung, where they see antigen again in the context of type 1 interferon and IL-15 to become cytotoxic CD4 T cells. And to put this more in the bigger picture, we see that it's not just a requirement for cytotoxic CD4 T cells at this point, but also for another specialized CD4 subset that is T follicular helper cells, where we see that CD4 effectors need to go to the secondary lymphoid organs, where they need to see antigen locally in that secondary lymphoid organ, which is they need to see antigen in the DLN to become DLN TFH, which is draining lymph nodes, or they need to see antigen in the spleen to become spleen TFH. And these required CD28 co-stimulation as well at this time point to differentiate our T follicular helpers, which could help B cell responses. And we had also found that CD4 effectors needed to see antigen again at this time point, just 6 days after infection, to most effectively form long-lived resting memory compartment. So we think that there is an effector checkpoint governing the transition of these CD4 effectors into more specialized subsets of CD4s, be it CD4 memory or cytotoxic CD4 T cells or T follicular helper cells. And we think that makes sense because you don't really want the generation of these more specialized effectors unless you need them. And the presentation of antigen beyond 6 days tells the immune system that, hey, there's still danger, there's still influenza infection, so we need to do more to clear this virus. And that's when you see the development of the more specialized effectors. Whereas if infection was cleared early with just Th1 effectors, you don't see the generation of the more specialized subsets. And that also makes sense because these are all potent CD4 effector subsets which have been implicated in various autoimmune diseases. And these findings also have implications for vaccine design, where if you want the generation of these more specialized subsets of CD4s, including actually long-lived CD4 memory, then you need vaccine to not just present antigen during priming at the initial phase, but you need more sustained antigen presentation as these CD4s develop into effectors to be able to transition to these more specialized subsets, which is why we think all these mRNA vaccines do a good job because of their sustained antigen presentation. And this also tells us that the route of immunization is important for where you want these subsets of cells forming, as well as adjuvants if you want these signals that infection traditionally generates. That's important for the development of these subsets. And for what this project has led to, I'm very excited to be starting my own group at Stony Brook Medicine focusing on respiratory immunology. And what the findings from this have led to is that these cytotoxic CD4 T cells that we showed need IL-15 and local antigen are able to go on to become long-lived tissue-resident memory in the lungs. So we're really excited that we may have found a precursor for this long-lived CD4 TRM, which both in humans and mice have always had this cytotoxic signature. And we've always wondered why CD4 TRM in the lungs have a cytotoxic signature. And that may be because these TSCTLs are an important precursor for the long-lived CD4 TRM. And something else that I'm looking to do is really, are nasal immune responses also important? And can having strong nasal immune responses reduce the burden of immunity in the lung? Where maybe if you clear responses, less of virus reaching the lung, so the lung has to do less, so there's less lung immune responses and less lung pathology coming from it. And maybe if you have an immune response in the nose, even before virus goes to the lung, you can also reduce transmission. And we're also looking at how these responses, including B cell responses, how they vary with age and what we can do to induce effective immune responses in the aged as well. I wanted to thank the Swain Lab where I've done this very exciting work. My mentor, Susan Swain, the graduate student, Alan Wong, who began this project with me, and a very talented research associate who helped me through these projects. Yeah, I would be happy for any questions and thoughts that you guys have. And thank you, Hannah, for this invitation.